Let me talk about question 6 of exam 21. This is an example of a problem where there's lots of different issues, a lot of genetic issues, a lot of biological issues. So the question is, always read the question carefully. Make sure you understand the question, because if you don't understand the question, you're unlikely to arrive at a reasonable answer. You have been hired, right? You have taken genetics and you're hired for a job because you've taken genetics. You've been hired to design an Ames test. An Ames test is named after Bruce Ames of UC Berkeley, who developed this test. It's a very elegant test, very sensitive test, very inexpensive test, and it's widely employed. Test for mutagens. Notice it's a test for mutagens. It's not a precise test for a carcinogen. It's a test for a mutagen. It's a test for something that causes a mutation. Likely anything that causes a mutation causes cancer, but nonetheless the test is specifically for a mutagen. So you've been hired to design an Ames test for mutagens that cause A to T substitutions, A to T mutations. So an A to T mutation specifically, because we can design these tests that detect specific kinds of substitutions, specific kinds of mutations, insertions, deletions. In this case, we're looking for something that causes an A to T mutation. You choose to construct a histidine auxotroph. So histidine is an essential amino acid. Although a normal bacterium would probably be prototrophic, meaning it can live off of elements and a carbon source like glucose. And we have what we call auxotrophs, those bacteria, those organisms that require a specific supplement in order to live, because it's lacking some gene in the biosynthesis pathway of that particular metabolite, in this case histidine. So histidine for us is an essential amino acid. For most bacteria it is not. So wild type bacteria, typically one we use Salmonella for this, would be able to live on minimal media, but we can construct a mutation in a specific gene along the histidine biosynthesis, perhaps His3. In this case, it's a hypothetical His gene involved in histidine biosynthesis, such that this bacterial strain will not grow on minimal medium, but needs to have supplement with histidine. It's a very important point here, because this way we can distinguish between wild type bacteria that will grow in minimal media, those bacteria that have a mutation so that they will grow only on histidine supplemented media, and those revertants that are able to grow on minimal media. So we can see very rare events, 1 in a million events. So mutating the His-X gene, a hypothetical gene, or could be a specific gene, but I've come up with an arbitrary sequence, such that it has an early stop codon. So we're told to use an early stop codon, although it's possible that a specific substitution amino acid would cause the gene not to work. A stop codon is a pretty clear way to avoid a functional enzyme that can revert to the wild type sequence upon a single, this is typical. If we're looking at mutations, we usually look at single mutations in isolation, so that we can understand their function. A single AT substitution. So what do we want to do? We want to take the wild type gene shown below, and we want to find where can we cause a single mutation such that an A to T substitution will revert it to wild type sequence. So we have something that will not grow on minimal media, and if it reverts, even a very small number of bacteria revert because of this mutagen we're going to be testing, or whatever mutagen we're testing, we can see that very rare event by growth on minimal media, by counting revertants. So use the 5' end of the coding sequence. Coding sequence, be careful, don't get confused. Template, non-template, coding, non-coding, plus, negative, positive. All of these you can get confused about. Be clear about this. In this case, the coding sequence is the strand that, if it were RNA, would be equivalent to the transcript that is translated. So it's 5' to 3' amino to carboxy sequence of the wild type histidine shown below to indicate precisely. Indicate how? Circle it, draw an arrow precisely. That means that base pair or base pairs in the DNA that would serve this purpose, if any, the enzyme sequences. So we have amino, met, arg, pro, lu, ti, lys, cis, lu, lys, threonine. So in single letter, met, arginine, proline, leucine, tyrosine, lysine, cysteine, leucine, lysine, and threonine. Doesn't matter. And the DNA sequence representing it, notice that this is just a fragment, so there's all sorts of other things missing, and we're only shown one strand. This is it's DNA, so it's double-stranded. We could draw the other strand there, 5' to 3' in the other direction, but it's probably not worth the time to do so if we were thinking clearly. Nonetheless, the single strand is shown 5' to 3', and the other strand, its complement, is assumed. Okay, so what are we looking to do? This is a wild type sequence as a functional. If there's a stop codon in this region, then the enzyme won't be functional. So we want to find where can we place a stop codon such that a single A to T substitution will turn it back into this. So we have to think a little bit oddly here. So what do I'm looking for? I'm looking for a stop codon. So that would be a TAA would be fine, or a TAG would be a stop codon, or a TGA. And notice those are all in the coding sequence. So if I want to keep in mind the other strand, which is not shown, that's a C, T, A, T, C, A. And I want to find something that if there is a T to, an A to T mutation, substitution, that it will turn something that's a stop codon back. Well, an A to T, considering that DNA is double-stranded, an A to T is the same as a T to A in the other direction, right? 5' to 3', then, right? Do you see what I mean? 5' to 3', I would have an A, and the other direction, its complement is a T. So if there is a to T mutation, that is essentially the same as a T to A, because if it's an A to T in this direction, it's a T to A in the other strand. So A to T is equivalent to T to A, because if the other, if there's a mutation in the top strand, the shown strand, well, that's equivalent to the converse mutation in the other strand. So what I want to do is look for any place I can take one of these codons and change an A to a T or a T to an A to generate a stop codon, such that when the A to T mutation of interest occurs, we regenerate wild type. So I want to know where can an A to T mutation or TTA mutation cause a stop codon so the mutation of interest will revert it. Okay, so if I'm looking here, so really what I'm asking for is which amino acids here, which codons are a single TA substitution away from a stop codon, TAA, TAG, TGA. So I can see that, well, ATG, no, I can't turn that in a stop codon with a single A to T mutation. AGT, yes, look at that. If this were a T, so if I made that a T, TGA, it would be a stop codon. CCA of proline, no, I don't see something. TTA, that could be a TAA. I'll write out the stop codon here to make it clear. Here, I see a stop codon here. There's also a stop codon TAG, but that's not related to a TA. I want something that's a T to A. A to T, which is T to A in the other strand. A to T, T to A here, T, A, G, here, T, G, A, here, T, A, G, here, T, A, here, I don't see anything. So that means if this A were changed to a T, there would be a stop codon. If this T were changed to an A, there would be a stop codon. If this T T were changed to an A, there'd be a stop codon. If this A were changed to a T, there'd be a stop codon. If this T were changed to an A, there'd be a stop codon. And if this T were changed to an A, there'd be a stop codon. And if this A were changed to a T. So all of these places, I can change the A to a T or a T to an A. A stop codon would be introduced such that when A N as written, so when there's an A to T mutation, substitution, in either strand, either the coding strand or the non-coding strand, we would revert to wild type. That's what we're looking for. So how would this work? There's a very powerful test here. We have a means by which we can take something that we don't know whether it's mutagenic or not, maybe some new compound that has interesting properties in medicine for human use or food or in the food chain for animal use, whatever. And we want to see if it's safe for the human use or the human food chain. So what we can do is we make a construct here with the stop codon introduced. We take that compound. Typically we mix it with liver extract, ideally human liver extract, but that's not so easy to come by, so rat liver extract. That allows any pro-mutagen, something that's not mutagenic by itself, to be converted by our metabolism, liver enzymes, into something that's going to exist in the body, into the final mutagen. And we have compounds that are not very mutagenic, but after a liver tries to detoxify them, ironically makes them very toxic. That includes some very classic mutagens such as benzopyrene, one of the first identified, or maybe the first identified mutagen or carcinogen in chimney sweeps is found in soot from burning wood. And one of the most toxic, most carcinogenic compounds, aflatoxin, which is found in mold, typically moldy peanuts, and the, cytochrome P450 enzyme tries to oxidize it, to detoxifies it, makes an epoxide, which reacts with the DNA and is very mutagenic. So one would take this construct we've made, we've introduced a single substitution so that we have changed from a wild type functional His gene, from a wild type prototroph, as it were, the prototroph, and turned it into a His oxytroph. Oxytrophs are very interesting for biological purposes. We've turned it into this. Now we mix this bacteria, expose it to this compound. After this compound perhaps had been incubated for liver enzymes, and we plate it then on minimal media and on His media. And we can see in His media the total number of bacteria, that we have, and by plating on minimal media, so we compare minimal media to His supplemented media, and we see what the fraction or percent viability is, the percent revertance. So there's going to be a natural rate because of mistakes in polymerase fidelity, or from other sources of mutagen, whether it's cosmic rays or oxidation, whatever, that we're constantly exposed to. And we can see whether this rate is above that natural reversion rate, and we can see to what extent something causes mutation in this context, in bacteria, Salmonella. And you might say, what relevance does this have to human carcinogenesis? Well, if it causes mutation in bacteria, it's very likely to cause mutation in humans, and if it causes mutation in humans, it's likely carcinogenic. And this is a very, very sensitive method, and it's a very, very cheap method. And so it certainly makes sense to test anything before it's exposed to humans here. Otherwise, it might not be until animal testing, which is extremely expensive, or human testing, in which case it's already gone to market or through clinical trials and a lot of people involved, to see something. Something may cause a significant rate of cancer, at least enough that the regulatory officials would not allow it to be exposed to humans, and it wouldn't show up until it's been exposed to a lot of humans or gone through very expensive animal testing. So this is a very useful test to eliminate things. So let's look at this again. So I'm asking, where would a T to A mutation be useful to generate a stop codon that we could use for this test? So that is that base pair, this base pair, this base pair, and we would only use one at a time because we want to look for rare examples. We don't want to look for double mutation. So any of those positions would work, which means our answer should be all of them. So 1, 2, 3, 4, 5, 6, 7. There may be some other answers possible with some clever things that I don't want to get into, but this would be the answer that I'm looking for.